Hello everyone, welcome back to our YouTube channel AgriEdit. So after a very long gap, after completing the genetics, plant breeding, plant pathology, I am back with the PYQ discussion series of the plant sciences 2024. But today I'll be discussing seed science and technology. Not only the question discussion, we'll be also discussing all the relevant options, all the related options that are given in the question, and we'll be also revising whatever concepts that you have learnt. Okay, so after watching this video, many things will be clear from the seed science and technology. So this will be much more useful in your exams. And if you talk about the number of questions that is asked in the exam from the seed science, there will be around 5 to 10 questions that comes every year. I'll be also highlighting like what are the important areas which you have to look for if you are preparing for the plant sciences for the 2025 and in the upcoming years. So stick with me until the end of the video, and if you get any value out of this video, I would request you please like, comment and share and subscribe our YouTube channel. So let's get started. So very first question, again it started with the assertion and reasoning type. So let's start. So assertion says the foundation seed is a progeny of a breeder seed and is a first generation of a seed produced after the breeder. So reason is saying the foundation seed is primarily used for the research purpose only. Okay, so let us just first look at the assertion. So here it is saying that foundation seed FS, it is the progeny of the breeder seed. So this is true, and it is also called as first generation of the seed produced after breeder seed. Yes, so if you know like generation system of seed multiplication, it starts with the breeder seed. Although we have nuclear seed, but it is not considered under multiplication. And here this breeder seeds to foundation seed to certified seed. So this is a general classification. So here this sentence is saying that the first generation of a seed produced after breeder seed. So if you look at this, so this is obviously the first generation after the breeder seed. So if I have to talk about certified seed, then this is the first generation after foundation seed, and it is a second generation after breeder seed. So this is how it goes. So for according to this is right, but whether this is explaining the assertion reason, like whether the reason is explaining the above statement or not, that we have to look for. But before that, whether this reason, the statement which is given under the reason, is true or not, let's see that. So it is saying the foundation seed is primarily used for research purpose only. The word primarily is important. So according to what you have learned from the seed certification chapter, so we know it starts with the breeder seed, goes with the foundation seeds and ends with the certified seed, which will be certified by the state seed certifying agency, right? But why do we go for breeder seed to foundation? So do we go for this first generation just for the research purpose? No, we actually produce the foundation seeds because we need certified seed. So we want to produce the certified seed, which will be certified by the state seed certifying agency. So that's why this is the intermediate stage. For this reason, this is entirely wrong. The reason is wrong here, which has used the term primarily, because primarily we use for seed multiplication, not for the research. If it is specific to research, it is mainly the breeder seed. In some of the sources, in some of the books, you might find that foundation seeds are used for research purpose, but remember, it is not primarily used. So that is how like they can confuse you in the exam. So please make sure that all your concepts are very clear here. We are going from the breeder seed to foundation seed to certified because we have to produce the seeds or for the seed multiplication. For this, the answer is A is correct and R is wrong or incorrect. Okay, the next question. So before that, I have also included some of the important information regarding the same. I'll explain this. So what is important for your exams? So here, this is not nuclear, this is nuclear seed. Other than what I had discussed in the question, what other things that you have to remember? See, the nuclear seed and the breeder seed, who is going to produce this? So these are being produced by the producing breeder or the sponsored breeder. Producing breeder or the sponsored breeder. Okay, and where they are going to produce this? So they are going to produce this in SAUs, the research farms of SAUs and the ICAR institutes. See, everywhere we read that the breeder himself produces the breeder seed, or the nucleus seed is produced by the breeder himself. But that doesn't mean that they will be doing it on their own land. They will be doing it on the ICAR institute lands or the research farms of state agriculture universities. That's the simple thing, right? This is what can be asked. Okay, who is going to produce this breeder seed and nucleus seed? So who is going to produce it? It is either the original breeder or the sponsored breeder. And again, if I use the term breeder, breeder does not always refer to the individual. See, it is not always the individual what we refer to. It can be also the institute. For example, if some institute has produced some variety, then the seeds of that variety, the initially produced, it will be the breeder seed, and the breeder is now institute, not just the individual. So that means it can be individual or the institute when we refer to the original breeder or when it refers to the sponsored breeder. Fine. So this is about the first stage. Next, once it goes to the breeder seed to the foundation stage, so who is going to produce it? So from this stage, remember, this is under what? This is under state seed certifying agency, and the as the name suggests, it is state seed and it is directly controlled by the state government. The agency is notified by the state government and the license is given by the state government, and it is under state government, which is the one which is starting or which is producing the foundation seeds and the certified seed. And where it will be produced? Again, it will be either in the ICAR institutes or it will be the SAUs. Along with the SAUs, it will be like NSC or National Seed Corporation, and it can be like state KVKs in different states. So these are the main areas where the foundation seeds will be produced. Along with this, it can be also produced by the private companies, recognized private companies which holds the permission from the agencies can also produce this foundation seeds. And the last level, which is certified stage 1 and 2. And I have clearly explained all of these things regarding certified stage 1, 2, why we are going it for 1 and 2. Please make sure that you know about these things. If you are not well aware of the seed certification chapter, even in the YouTube, like I have uploaded the free videos which are available for everyone. The links will be available in the description box. You can just check this video. And totally for seed science, like I have uploaded all the seed science videos which are just 16. See, if you watch the 16 video, I can guarantee you that you will be able to solve most of the questions that is asked in the exam of ICAR PG. So make sure at least to cover the 16 videos if you are not a student of master course. But if you are a student of master course, you already know, like I have explained it clearly, what is this 1 and 2. Okay, and this is about the 3 categories. And if I have to list out, this is for the foundation seed and this is for the breeder seed. And here I told you, like NSC, this is like State Farms Corporation of India, and SSCs, State Seed Corporations, Agriculture Universities, SAUs, and also the ICAR institutes. All of them are involved in the production of the foundation seeds. But when it comes to the breeder seed, so these are the 2 main institutes or the organization. And when it comes to the certified seed, along with all of this, progressive farmers will be also involved in the production of the certified seed. So that is the one extra thing that you have to remember here for the certified seeds. Okay, and if I want to recall the color, if you know about the color, this is again not nuclear, it is nucleus seed. So what is the color of nucleus seed? What is the color of breeder seed? What will be the color, tag color of foundation seed and the tag color of certified seed? And lastly, we also have the truthfully labeled seed. I want all of you to comment on this video if you know the answer. This is a very basic and the most frequently asked question in the ICAR PG exams, in any other exams wherever the seed technology syllabus. So I want everyone to comment on this video like the answer to this question. I'll be very happy to see your comments. Okay, that's it. Next one. I hope like this is very clear. Next, the question says the fertilization is an important event in the angiosperm which involves the several critical activities. Below are the few major steps in the fertilization in the plants. Arrange them from the beginning to the latest systematically. So this is again the question from floral biology. The first question which we saw, it was the question from the seed certification, and now we are getting the question from the floral biology. So here we have to just arrange it from the beginning to last. It's very easy question. Whenever you get such type of questions which are mostly related to the 11th and 12th and which are very basic, you should never miss it and you should never make any mistake on it. So first thing, it is pollen germination, anthesis, fertilization and penetration of the ovule. So if you know what is anthesis, anthesis is like opening of flower. So fertilization, pollination, everything happens only after opening of flower. So we can we keep it as first step, right? So once this is open, the pollination is occurred. So once the pollination is done, and here once the pollen falls on this stigma, so it travels till to the ovary, right, through the style, if you remember. So that means it is like called as pollen germination. So once the pollen is germinated, so it will penetrate to the ovule, forming the pollen tube, and once the tube is formed, finally it reaches the ovary and fertilize itself. So that C will be the last step. In the whole option, it's very easy that you can see that the C is the only option that we have is C, which has C at the last. So we can easily mark this as a right option. So this is how much easier some questions will be in the exams, where you need not to invest much of your time. Just see the options, see the questions, try to relate with them, and you will get the answer for sure. Okay, and this is B and A and D and C. Same thing, the pollen has fallen, and here a pollen tube has developed. At the last, it penetrates the ovule and the fertilizes the ovule, egg cell. That's it. Okay. Next, so now this is the question related to seed testing. Now directly from seed certification and solving the floral biology question, we came into the seed testing part. Now this is saying about the 4 different kind of tests. One is growth test, second one is tetrazolium, third one is electrical conductivity test, fourth one is imbibition test. See, I'll tell you, like, if you get any questions in the match the following for your ICAR PG exams, take it as a sure shot. You have to compulsorily attend it, and you have to just see it properly, even if you don't know anything about the match the following, whatever is asked here. So apply your mind, keep calm, be calm. So just see like the options and the questions. If you are able to make at least 1 or 2, the answer is here. You will be able to get it. So make sure to try it out. For example, here you can see growth test. Growth test, everyone knows it is like GOT, right? GOT, why it is done? It is for genetic purity, or can we write it as seed purity test? So that is done. And tetrazolium. So tetrazolium, everyone knows this is TTZ, also called as TTZ, and it was given by Lacon. And this is a seed viability test. And what is seed viability, if I ask you? This is a test which says whether the seeds are alive or whether they are dead, right? So this is for the seed viability, the second one. The next one, the electrical conductivity test. So if you recall or remember, like, what does it test? What do we do in this test, or how this test is conducted? You will be getting the answer. See, it's very simple. In electrical conductivity test, we see the amount of leakage of solutes from the seeds, right? So if the EC value is high, then what does it mean? It means the seed vigor. So this is the estimate of vigor, this electrical conductivity test. That means seed vigor is low. Why the electrical conductivity is high and seed vigor will be low? That because the I value refers to high amount of leakage. If there is a high amount of leakage, that definitely means seed is actually losing its power or potential to grow. So that's why this is for the leachate discharge. And the last one, the imbibition test. So this is for the coat permeability, where we do the imbibition test whether the seeds will be able to like intake the water or not. It depends on the how much permeable the seed is. So this is about the imbibition test. So this is how we can easily answer such type of questions. So to recall all of these things from the seed testing, I'll just give you a brief idea. From seed testing part, like, we are too much worried. Like, we are always worried, like, we have so many different types of tests and we fail to remember each one of them. And when it is asked in the exams, everything feels very much confused, right? So I'll try to classify all of them in detail incorrectly, because if you remember, you have phenol test, you have NOH test, you have like TZ test, you have embryo excision test, and you have the phenol test. So there are so many different number of tests, like you have GOT test, right? But where these tests are used, for what purpose they are used, we are always confused. We will not be able to classify it correctly. But after watching this video, you will be able to do it. I'll try to simplify this for you. First thing, like, let us just recall the number of tests in your mind. See, it's not very complicated. It's very simple. In the seed testing, these are the number of tests you have. One is to check the physical purity of the seed, and second one is like to check the genetic purity of the seeds, and the third one is testing the vigor. That is, vigor is what? It is the potential of the seed, how much potential the seeds are. And the fourth one, seed health, whether the seeds are healthy or not. And the fourth one, germination, how many seeds, like if I take the 100 seeds, and how many seeds out of 100 will be able to germinate. So that is what we do in the germination test. And next, the viability test, whether the seeds are, as I told you, it is alive or dead. So that is what we do in the seed viability. And lastly, we have moisture test, where it estimates the amount of moisture or the water that is available in the seed. Okay. So let's start with the physical purity. So if for testing the physical purity, like how many number of tests are available? See, it's very simple. We have one simple test which is called as table examination. So what do we do in the table examination? We take specific sample, and here in all of the tests, if you ask me like what is important, you have to remember the working sample for each of the test, the number of tests that are available for each of this, so that is important, and the methodology, how we are going to do it, and giving out the results, how the results are expressed. So these four things are important from all the tests that I have mentioned here. So starting with this one, this is just a table examination where the specific working sample will be taken, it is spread across a table, and each one of them will be individually examined carefully on a table, and that is how we will be able to classify it. And again, along with this, in how many fraction, like inside the methodology, we have to know like how many fraction it is divided. So what I mean to say is, for example, if we are doing the purity test, it is divided into three different fractions. What are they? They are like pure seed, and it is like inert matter, and the last one is other crop seed, and this is the international classification. If you talk about the Indian classification, along with these three, one more thing adds, which is the weed seed. If you remember like what I have discussed in the class, this is the four. So this is according to the ISTA. What is ISTA? Again, ISTA is International Seed Testing Association. And when it comes to the Indian system, so we have four different categories under the physical purity testing. So this is about the physical purity. And next, about genetic purity. Genetic purity, we have several tests here, but remember the one most important thing, this is mandatory, which is GOT. Why do we always pronounce GOT? Why the other tests are not much pronounced as much as GOT? Because in India, to test the genetic purity, we have only adopted grower test. In India, to check the genetic purity of the seeds, we are only using the GOT, which is grower test. That's why it is more pronounced, and this is why you are more familiar with this test compared to any other test that you have for genetic purity. Because if I ask you simply like, see, tell me the list of tests that you have for genetic purity, the one and only thing that you'll able to remember is GOT. I know, because this is because of the reason that in India for certification only GOT is used. And next we have vigor. So vigor is the most complex among these tests. Complex in the sense like it has multiple number of tests that you have to remember. That's why it is complex, nothing else. It is also easy, but to remember you have to remember the multiple tests. And seed health testing, so there are not many, and it is not entirely included in your syllabus. So whatever that you learn from the plant pathology, that is more than sufficient here. You need not to read anything extra. And germination test, for testing the germination, we use mainly three different materials. One is like paper can be used, second one sand can be used, and third one soil can be used. But soil is not recommended because it can be phytotoxic. So generally the paper is used for the germination test, okay, whether it is a seed testing lab or for the research purpose. So we mainly use the germination paper to test the germination. Either the petri plates will be used, like there are again the number of methods, like top of the paper, between paper, pleated paper. So these methods are generally used, and the substrate which is used here is paper. And next, the viability. And when it comes to the viability, just like the genetic purity test, you know the one major test, which is it? It is just the tetraazolium test, which we are mostly familiar with. So if you are familiar with tetraazolium test, I would definitely recommend you just try to read each and everything about the test, starting with the principle of the TZ test. Read everything. That's very important, and it is the kind of seed viability test. So when I come with the marathon series on the seed technology, I'll try to cover this. And if you need the marathon series for the seeds and technology and for the genetics, in which in a one go I'll start from the starting and end with the completing the syllabus in one go. If you need that series, I want you to comment on this video. Okay, I would like to see, I would love to see these comments. So based on the number of comments, number of engagement that we get, so we would be definitely motivated to bring a lot of quality content for you people for your exam. So lastly, we have moisture testing. Again, in the moisture testing, there are many tests, but the one routine test that you have to remember for your exam, it is like hot air oven method. If you have heard about it, hot air oven method. Okay, and again this has a two different temperatures, which is low constant oven method, LC, low constant temperature oven method, and high constant temperature oven method. I'll tell about this in the upcoming slides. So this is the simplified form of the seed testing procedures or seed testing types that you have to remember, starting with the going for the physical purity, it is table examination. Going with the genetic purity test, it is mainly the GOT, and next coming into the germination, it is the top of like paper method, in which like we have top of the paper, between paper and pleated paper. And next coming into the viability, it is T-set test. And the vigor has like different categories. Under different categories there are several tests that we have to remember. I'll just show it in the slide. And whatever information that I'm giving you orally, I have also compiled this everything in the PDF, in this PDF, and you already know like what you have to do to get this PDF. These PDFs are like whatever we are discussing from the PYQs series, starting from the genetics, all the PDFs that are discussed in the YouTube, it will be provided to each one of you if you need this PDF. And if you need it, you can just fill the Google form that is available in the description box. And if you have filled already in the previous videos, then no need to fill. You will be already getting it in the drive. Okay, you have to just check your drive. So after watching this video, just check it. So the same drive, you will be also getting this PDF. Okay, in which like all the informations are compiled. So here, this is regarding the genetic purity which I have listed. So now, as I told you, the methodology is important, but do not go for the methodology of all of these tests for genetic purity. Only the methodology of GOT, like the working sample and the procedure, those things are important. That's it for only this test. For all other tests, just remember the names. For example, we have phenol color test, it is specific to wheat, you have to remember this. And we have modified phenol, it is also for the wheat. For example, we have seed peroxidase test, it is peroxidase test, it is for specifically soybean. And KOH bleach test for sorghum, ferrous sulfate test for paddy, NaOH plus potassium dichromate test for the pea and wheat. So these examples can be asked in a match the following. That's it, nothing more. You have to remember the test name and you have to remember the crop. That's it. And lastly, we have alkalites for lupins. And similarly, we have biochemical test, we have examination of seedlings. Here we have 4 different types like coleoptile color, hypocotyl test, and the cotyledon color test, fluorescent test, and that are specific to certain species that you have to remember for sure, and it's compulsory. The questions can always come from this part. And lastly, that what I mentioned, it is under the third category, which is field plot test, that comes the GOT. Okay. Next, so what is this? This is germination test, which I explained earlier, where the growing media is paper mostly, and in that we have top of the paper, between paper, pleated paper, and other than that we have sand or organic matter, and lastly soil, but which is not generally recommended. Okay, next. So the seed viability. So I told you in seed viability we mainly use, we are aware of the tetraazolium test. So make sure you know everything about this tetraazolium, starting with the principle plus the working sample, the methodology or the procedure. So each and everything is important. Anything can be asked from this part. And other than this tetraazolium test, which is most common test, which is used for measuring the seed viability for tree species, remember, for tree species we have 2 different tests, which is embryo excision test and the other one is X-ray test. So this is for specifically the tree species. Okay, remember, this is for the tree species. And I also told you the reason why it is tree species in the class. So if you remember it, just comment on this video. And next, it is not actually included in that viability test. This is the one more different test which we use. It is like detection of mechanical damage, and here the ferric chloride test is used for, if in the exam they ask like which test is used to detect the mechanical damage in the seeds. Remember, it is ferric chloride test, where the injured seeds upon exposure to this ferric chloride turns black. That's it. Okay. Next, so vigor test. I told you the vigor test. Again, this vigor test can be broadly classified. They can be broadly classified into 4 types. One is physical test, second one is performance test, third one is stress test, and fourth one is biochemical test. In the physical test, we are just trying to measure the seed size, for example, seed length, seed width, seed thickness, and seed roundness, shape. So based on all these characters, we'll categorize whether they are vigorous or not. And coming to the performance test, based on the seed vigor index 1 and 2 and speed of germination and uniformity of germination. So all this will be the performance test, and if you are not aware of that seedling or seed vigor test 1 and 2, please make sure to read it once. Seed vigor index 1, and there are 2 different index for measuring the seed vigor, which is named as seed vigor index 1 and seed vigor index 2, and these 2 are important. Okay, I'll tell you like what are these exactly. And next we have stress test. By the name itself indicates like what is stress test. Like we are trying to give the stress and trying to calculate or trying to know the potential of the seeds. So here it has like cool test, we have cold test, and most commonly known like Iltner test, we have paper piercing test. So all this comes under the stress test. So that is how you can remember. See, we like usually know many of the tests, like we know about cold test, we know about cool test, we know paper piercing test, we know electrical conductivity test, we know accelerated aging test, but we fail to classify. If you fail to classify, then it's very difficult to remember, and also it is very difficult to link with their principle, and also most importantly to the test. These are the categories under seed vigor test that you have to remember. Next, the last part is like biochemical test. What are the different tests that we have under biochemical? You can also include TZ test, you can include electrical conductivity test. So again, do not get confused that sir, TZ test though like it is for the seed viability, why you are considering this under seed vigor? So TZ test, according to the staining properties, it can also give us some insights about the vigor of the seeds, where the highly stained seeds can be considered as highly vigorous, little stains based on the staining properties, based on the concentration, seeing the differences in the staining, how much staining the seed has been undergone. Based on that, we can classify it as highly vigorous, medium vigorous, and low vigorous. Any number of categories we can make, okay, in a complete range. So that's why the TZ is also included in the vigor test. As I told you, like performance test, like first count, speed of germination, everything is included. So just have one look at it, and again, nothing to worry. Do not like try to read all of them in detail. The procedures, everything is not important here. So whatever is important, I have already given it in the slide. Just make sure to remember that you know about the cold test and for which crop it is used, and you know about the cool test and for which crop it is used, and brick dowel test and paper piercing test, like you know about the test and for what purpose they are used, and if there are any specific crops, you have to remember the crops. That's it, nothing more. Do not feel it complex. I am trying to make it simplified, so feel it simplified, not the complex one. And next, the stress test. In the stress test, similarly we have cold, cool, brick gravel, so same thing here. And next we have the biochemical test. So first one, this was asked in one of the previous exam. So here try to know simple things about this kind of test. Here GADA test, it is like glutamic acid decarboxylase test. So what is this decarboxylic acid test? So here this is the enzyme which is actually present in the seeds which are active. Okay, if the enzyme activity is more, if this enzyme activity is more, that means that the seeds are highly vigorous. How this enzyme activity is tested? See, in this test glutamic acid is usually treated, and this enzyme which I talked about, this actually converts this glutamic acids and releases CO2. Okay, so if the CO2 emission is high, then what does it mean? Does it mean that it is highly vigorous? Because CO2 emission will be high only if the enzyme concentration is high or enzyme activity is high. So that's why this is highly vigorous. So this is under the GADA test. Next we have tetrazolim test, you already know about it, and respiration coefficient test and ATPase level test and the electrical conductivity or electrical conductivity, both are same, do not get confused. It is one and the same, where we try to calculate the leakage of, so where we try to calculate the leakage of solutes. So I'm repeating it again. If the EC value is I, that means the seeds are less vigorous. Okay, remember this point. This might confuse you if you do not remember it. Okay, next, the most important one that you have to remember from the seed vigor test is ISTA accredited test. So what is this ISTA accredited test? If you know about the ISTA is the international organization which provides the procedures for the test, for the seed test, right? So this has recommended some of the tests for calculating the seed vigor. So it has recommended this test and standardized this for the certain crops, that they are mentioning that ISTA has mentioned that for this particular crop, this test is most valid for calculating the vigor. So for example, accelerated aging test, it is most useful for testing the seed vigor in case of soybean, and control deterioration test for brassica, conductivity test for chickpea, garden pea, French bean, radish and soybean, total of 5 crops. And next, like tetrazolim test for brassica and radical emergent test. So this is for wheat, maize, brassica and the radish, total 4 crops. And this is the one what you have to remember, that how many ISTA accredited seed vigor tests are there. There are total of 5 ISTA accredited tests, and again in each of the tests how many crops it has been used for, that you have to remember. This question can definitely come in the exam, so make sure you know it. And I told you about the seed vigor. So seed vigor, how it is calculated, it is called as seed vigor index 1 and 2 and seed vigor index 1. So this is like mean seedling length into germination percentage, first one. And second one, vigor index 2, it is like mean seedling dry weight. Here it is dry weight in the second index 2, in the index 1 it is seedling length, which is in centimeter. Also remember this unit, it is in centimeter and it is in milligram. Dry weight into germination. Common thing is germination percentage, but the difference is first one is like seedling length and the second one is the dry weight, and first one is in the centimeter and second one is the unit is the milligram. Fine. And last is about the moisture testing. So if we cover this moisture testing right now, that means we have thoroughly covered the seed testings of the different categories. This is how much simple it is. So we started with genetic purity, we covered germination, we covered physical purity, we covered the seed vigor, viability, and now on the moisture test. Okay. So again in the moisture test there are actually 3 different methods. One is reference method, the second one is practical method, and third one is routine method. So you just know the names of this test, nothing much in detail is important, you can entirely skip it, but concentrate on this routine test. So what does the routine means? So this is a test under the moisture testing which is actually used in the seed testing laboratory. That is what it means. So what is the method? It is hot air oven method or air oven method, which is again recommended by the ISTA. And again it has 2 different types: low constant temperature oven method, LCTOM, and high constant temperature oven method. So here the difference is in terms of temperature. In the low constant it is 103 degrees Celsius for 17 hours for less than 70% RH, and for the high constant temperature oven method it is 130 degrees Celsius for just 2 to 4 hours. And this is for the cereals, maize and oilseeds. And for cereals and the maize it is high constant temperature oven method, and for the oilseeds which cannot actually withstand the high temperature, it is done by the low constant temperature oven method. So for you in the exam, make sure to list out as many crops as possible into low constant temperature oven method and high constant temperature oven method. So this can be asked. For SRF it's must and should. For the ICAR PG exams, the chances are high that they will also ask for which crops we are using low constant, for which crops we are using high constant. So please make a list of this example, that will be useful for your exam. And the safe moisture for storage, I have also given you some information regarding the storage. If the seeds has to be stored, then cereals should be stored with the moisture content of 12 to 14%, and vegetables should be stored with the lower moisture which of 8 to 9%, and oil seeds with just 7 to 9% of the moisture content. So this is the moisture content. Also remember this, This is also very, very important. This covers entirely the seed testing part. Okay, let's go to the next question. The characteristic features of the monocot seeds include, they are asking about the characteristic feature of the monocot seeds. So here the seeds germinate bearing a short axis called as plumule and radical. So this is right, but also will classify whether it is true for only monocot or it is also true for the dicot. The first statement which you are saying, the seed germinate bearing a short axis called as plumule and radical, this is true for both monocot and the dicot. There is no change. Okay. Next one. The plumule and radical are enclosed in a sheath called as coleoptile and coleorhiza. So this is specific to monocots or it is for both monocots and dicots. So this is for monocots, and dicots usually have a seed coat structure, okay, for the protection, but the monocots generally have the coleoptile and the coleorhiza. The third one, the radical grows downwards and develops into the root system. This is true that radical is a part which goes downwards and develops into the roots, but is it true for only monocots or it is true for both monocots and dicots? So it is true for both monocots and dicots, and this is for monocots. I'll try to write it as mono, it will write it as dicot. Okay. Next one, the cotyledon of the seeds of the monocot crop sorghum is known as husk. So they are saying that cotyledon of the seeds of the monocot is called as husk. Is it true? So this is not actually called as husk, this is called as scutellum. So what is husk? It is just the outer covering of a seed, right? So out of this, what they are asking, characteristic feature of the monocot. The first one is a characteristic feature of monocot, second one is also a monocot, third one is also a characteristic of monocot. Again, do not get confused here. They have not asked you which of the following is the only characteristic. They have not asked about the only characteristic. They have just asked like which of the following is the characteristic of monocot. So according to this, A, B, C will be the right answer. Hope it is clear. And here I have tried to give the complete difference between the monocots and dicots. In every class like that I take, I always emphasize this. See, whenever you get two different things of a same concept, try to differentiate between them and try to make your own table. The questions will always come from there. For example, we have monocot and dicot, like based on the embryo, based on the root system, stem, leaf, flower, and the general characteristics. I try to differentiate it and give it in the table. This table will be actually beneficial for all of you. Just have a one reading of it and try to revise it from time to time before going to the exam. Next one, which of the following is the common strategy used for the seeds to survive the harsh conditions? Rapid germination, no. Deburial, no. And dormancy, okay. High metabolic activity. So this can't be the common strategy. The third one is the most relevant answer that we already know. It is a dormancy, which is being used as a common strategy to survive the harsh environmental conditions, right? Next one. So what is seed dormancy? Like I try to explain bit in detail about the seed dormancy. So seed dormancy, if you know, like it is just a temporary state of halt or temporary suspension of growth where the seeds will not germinate, right? Now, according to the types are again the most important ones for your exams. So dormancy, you know it. So even under the favorable conditions, because of either the seed coat structures or the internal structure of the seeds will not germinate. If that is the case, we call it as dormancy. That's very simple. But what are the different types we have? We have like paradormancy, we have endodormancy, we have ecodormancy, we have primary dormancy, secondary dormancy, scotodormancy. There are several different terms that are being used in the dormancy, but we have to make sure that what is the exact classification of it. This classification is very much important, not only to answer the questions but also important to remember it. If you remember the classification nicely, only then you will be able to remember it for a long period of time. Otherwise, if you just try to mug up, you'll forget very soon. So depending on the types, they are classified as based on location and based on type. So based on the location of the dormancy, it can be divided into two different types. I'm trying to give the simplest classification. So here it is like embryo dormancy, and the second one is seed coat imposed dormancy. So this is based on the location. What does it mean? If the dormancy is because of the structures of embryo, the location embryo, then it is embryo dormancy. Whether if it is because of non-embryonic structures or the outer coverings, then it is called as seed coat imposed dormancy. So this is under based on location. But what about based on time of induction? this can be based on time, it can be divided into primary and secondary. Okay, so the primary dormancy and secondary dormancy. And secondary dormancy, it is also called as induced dormancy. Okay. I have given the complete information regarding all of these terms, whatever I have told you. Like I just gave you the terms earlier, but in detail explanation is provided in the slides. If I try to break all of this and try to explain it, it won't be completed in like 1 hour. So try to read these things. Once we come for the revision series, like if you comment, if I feel happy, then definitely I'll put all of my efforts to come up with the revision series. Then we will discuss them in detail. But for now, for the exam, make sure you read all of these things. Seed coat, okay, and that's types of secondary dormancy, which I told you about: scotodormancy, thermodormancy, and complex patterns, and the classification. See, there is like 4 different systems of classification that are there, but all these systems are not important. Just see the terms once, but remember there is a classification from Baskin and Baskin. So that is the currently accepted classification for the dormancy. Try to remember that one. Again, not in detail, just the names, like into how many types that classification is made. For example, what I am trying to say is, like, it was in 2004, it has made into 3-tiered system, which consists of like class, levels, and types. And just try to know the 5 different classes of the dormancy classification. That would be more than sufficient. So if I have to talk about it, it's physiological dormancy, morphological dormancy, morphological and physiological, morphophysiological dormancy, and physical dormancy, and last one is combinational dormancy. So these are the 5 different categories under, or 5 different classes under the class. And the 3-tier system which I was talking about, it is class, levels, and types. But in depth is not required. Just try to remember this classification name, that it is given by the Baskin and Baskin, and it has a 3-tier system which contains the class, levels, and types, and starting it has a 5 different classes. That's it. Okay, so let's go to one more question, which is enforced dormancy. So where do you see this enforced dormancy? Because what I have discussed in the previous slide, I told you about 2 different types. One is based on the location. So whether it is like embryo, which is called as embryo dormancy, and second one, it was like seed coat dormancy. And based on the other one was regarding primary dormancy and the secondary dormancy. So this is what I have discussed in the previous slide, but we got a new term. What is enforced dormancy? So if you want to know about this enforced dormancy, and we have to go back and try to read out the other classification out of the 4 which was given. So here in the ARPER system, we have a term called as enforced dormancy. See, what the overall concept is very simple. Different people try to understand the dormancy in the different ways and try to give the different classification. So over the period of time, based on like the kind of work was done, so until today we have the latest system which is followed, which is Baskin and Baskin. But earlier to that, we had the other system. In one of the other system, this term was used, which is called as enforced dormancy. So enforced dormancy is something that the dormancy is induced. Like how it is induced? By providing the unfavorable conditions, which also means the quiescent stage. What is quiescent stage? How do you differentiate from the dormancy? If you remember, see, dormancy, it is like even under favorable conditions, it won't germinate. But quiescent is something which only do not germinate under unfavorable conditions, which is similar to the enforced dormancy. Okay, so this is how we can remember it. It is based on time. The earlier was based on location, which I did not write here. Yeah, it is time and it is location. Okay, I hope like the things of dormancy is clear. Let me just repeat like what you have to remember. First thing is the meaning of dormancy, that you have to know. It's very simple, where the seeds will not germinate. That is what dormancy means, even if they are kept in the favorable conditions. Next, the types, the classification of dormancy. When you talk about the classification of dormancy, like you have 4 different systems. Out of 4 different systems, the last one which I talked about, it is like Baskin and Baskin. So this was the one which is currently accepted, and according to this system, it has 3-tier system, which includes the class, levels, and type. Okay, and in the class we have 5 different class. That's what you have to remember. That's it. Okay. Next, let's come to the next question, which is about the seed type and the example. The match the following, again the very easiest one to answer. So orthodox seed, recalcitrant seeds, impermeable seed coat, and hypogeal germination. It might sound confusing, but I'll try to make it easy. First thing, out of this, can you see something which is related to the impermeable seed coat? So impermeable seed coat, does it means the hard seeds? Hard seededness is a problem of which family? Is it in the legumes? The problem is most commonly seen in legumes. So soybean is a legume. So here the direct answer here it is like 344. Just see in the option. So 344 is available in only one option. This is how much easy to crack the answers of the match the following. Still, I will be able to I will be discussing the other options, but for you, for these type of questions which are asked in the match the following, that is how you can crack it. Just try to find out one at least. Many times you will be able to come into the conclusion. The next thing, the orthodox and recalcitrant. Hypogeal germination. So if you remember the hypogeal germination, it is for chickpea, because dicots generally they have apigeal germination. But again there are exception. One of the exception, very important exception, is chickpea. So that's how we can correlate this hypogeal with the chickpea. And when it comes to the orthodox seeds, so capsicum is the orthodox seed, and recalcitrant it's coffee. But the coffee is not actually the recalcitrant. What is the category of the seed which to coffee belongs to? This is actually the intermediate seed. So do not get confused. Do not get confused in the exam. If they are not giving intermediate and they are trying to push the coffee into the recalcitrant, accept it. That's it. Because the recalcitrant behavior and the intermediate behavior are almost same, where the storage, they can't be stored for a long time. And if you talk about the timeline of this classification, first the orthodox and recalcitrant were discovered and they were classified and definitions were given, and in later to that the intermediate part was added. So there is still a lot of confusions or a lot of you call errors in the examples that I've given in the different scientific papers, but still remember, for now, if the options are only orthodox and recalcitrant, push it towards recalcitrant if the intermediate is not given. That's it, very simple. So hypogeal and epigeal, we got the new terms here that I wanted to discuss. One is hypogeal and other one is epigeal. What is hypogeal and epigeal, if you remember? So the hypogeal germination is the one where the cotyledons stay below the ground level or within the soil. Only the epicotyl emerges above the soil. If that is the case, it is called as hypogeal germination. So it is hypogeal germination. Okay. And if you see this in the figure, so here the cotyledon has come above the ground and hypocotyl has also emerged above the ground. So in this case, where the cotyledons come or emerge above the ground level or the hypocotyl is pushed outside, so this is the example for the epigeal. Now, what is important for your exam? Knowing the hypogeal and epigeal is most obviously important, but along with that, the examples. You cannot miss the examples of the hypogeal germination and the epigeal germination, the examples of the orthodox seeds, intermediate seeds, and the recalcitrant seeds, examples of the endosperm seeds and the non-endospermic seeds. All of these are like very commonly asked questions in the exams. So please make sure to make a table and try to remember all of these things as an example. And what is the shortcut for this? There is some shortcut for few, like for example, for Rabi pulses are all hypogeal except Rajma, and for all Kharif pulses they are epigeal except red gram. And along with this, sometimes like you'll get a crops, you will have no idea and you have never listened to it. If in case such type of questions are asked, remember the common trick that most epigeal germination is mostly common in dicots. Mostly the dicots are epigeal, but there are good exceptions like chickpea you have, but majority of the times the dicots are epigeal, and this monocots, the hypogeal is most common. For the rarest and rarest, like if you get in the exam in case, so remember this and use this trick. This will definitely work. Other than that, try to remember the other examples like gram, peas, cereals, bean, runner bean, red gram, and here you have soybean, beans, onion, brassica, castor, tamarind. So these examples you can't do it in a single day. that's very straight to you. So you can't remember all of this in a single day. Like you have to revise it every day or every alternate day, or discuss with your friends. So only then you will be able to remember all of these examples, the types, whatever that you have to mug up. It is not a single day process. Please make sure that you would read it today and leave it. That's not going to work. Even if you have made a very good notes of it, you have comprehensively covered each and everything, compiled the things, but you fail to revise it, then it would be out of your mind for sure. And in exam, even like if you see the options, sometimes you will not be able to remember anything. So please make sure to revise these things, keeping all of them at one side. Do not distribute it. And for examples for hypogl and epigl, I have tried to give the example for in case of vegetables too, which was missing in the previous slide. So make sure to go through this once. Okay. Next, impermeability. I talked about impermeability of seed, which is like hard seededness. So hard seededness, it is common in, I told you, one family. But what about the other family? So other than legumes, it is also common in the okra and cotton, that is Malvaceae family, and some species of Convolvulaceae family. Okay, just try to remember this if you are able to, because questions might always come, but at least know the meaning. In the mass of following, if they ask you about impermeability, like at least know the Leguminous family and the Malvaceae family. These two are very much important, and also like this is more familiar to remember this one. Okay, just try to remember these three families and the species. If not all the species, at least the families. Okay, next I talked about the storage behavior. What is it exactly, the orthodox, intermediate and recalcitrant? If you recall, if you are able to recall it and just answer yourself, what are these categories? See, these are the categories of the seeds which are made based on the storage ability. Based on the storage ability of the seeds, this classification was made. And how it was made? It was in 1973 by Roberts, who classified the seeds based on storage behavior into orthodox and recalcitrant. And later, along with the Roberts, there was one more scientist who is Ellis. So they classified the intermediate type in between the orthodox and recalcitrant. So this classification is based on the drying and the freezing tolerance. Simple, very, very simple. So this classification is based on whether the seeds are tolerant to drying, tolerant to freezing or not. That's it. So if the seeds are tolerant to drying and freezing, then it is orthodox seeds. If it is tolerant to both the conditions, that means it is like it can be stored for long period of time. That's a common sense, right? Next, if it is drying tolerant but not freezing intolerant, then it is intermediate seeds. And last one, if it is not tolerant to both of these conditions, then it comes under the recalcitrants, where recalcitrant seeds are the one which have initially the high moisture content, and the moisture content cannot be drastically decreased, and neither it can be freezed. Okay, stored under like low temperature condition. Both the things are not possible. So storage of this is very difficult. We have to go for cryopreservation. But in case of orthodox seeds, these are very easy to store. So this is about the orthodox, intermediate and the recalcitrant in the simple words. Other than that, whatever is there, you just have to go through it once. So that will be important for your exam. But the meaning I told you, it's mainly based on drying tolerance and the freezing tolerance. That's it. And the examples are quoted. The examples of for orthodox are many. The examples of the recalcitrants are moderate, but the examples for the intermediate are few. So try to remember the examples of this intermediate, and next for the recalcitrant. So whatever you remember for these both categories, rest it will be orthodox. Okay, and also the moisture content. For example, the vegetables or the seeds which have the high moisture content inherently while they are grown, remember they will be under the category of the recalcitrant, because they have already the high moisture content. When the water content is already high, it can't be drastically dried. So that's how you can remember it. And jackfruit, like many like get confused this with the intermediate seed, because I also know like in some of the books it is mentioned under intermediate, and also the coffee, like it might be have mentioned in the recalcitrant, but this is the very authentic. Like I have already checked the different resources and cross verified it, so you can remember this, but do not create any debate with anyone. read it yourself and keep it with you, and if asked in the exams, so this is how they will ask you, and this is how you have to remember and give it. That's it. Okay, the next one. Which of the following groups of seeds represented the orthodox seeds? So whatever we have discussed in the previous slide, the same question has been asked. Let's try to classify it. The first one, the coconut and mango. This is recalcitrant. We already know it from the previous slide. Rubber and cocoa. This is also recalcitrant seeds. And rice and wheat. Just by observing this, we can tell that cereals. So this is under orthodox seeds. And the last, the coffee and palm. It is under the intermediate category. So now they are asking about the orthodox. So which is the orthodox one? It is wheat and rice. So this is anyhow easy, because as soon as you see the cereals, anyhow you have to choose one option from the four, and they are asking to about the orthodox. Even before checking the other one, you can directly mark it as rice and wheat. Even though like you feel that okay, they are asking the examples, like how we can remember, even without remembering anything, if you just try to see the question, you will be able to answer it. No need, there is not even a need to remember the examples in some cases. You have to just apply your common sense. That's it. And how you will get that ability to apply your common sense? The more and more mock tests that you attend, more and more questions for you to expose. Like once you expose yourself to more and more questions, only then you will be able to develop that capability that you will think and you will try to answer logically using your common sense sometimes whenever you do not know the answers. So how to do it and where you will get the questions? You will get all the mock test questions which are prepared by the top qualified mentors from the IRA. So the mock test links are available in the description box. If you are preparing well for this exam, if you are prepared well, or still preparing for this exam, but if you have not yet attended any of the mock tests, I would seriously recommend you or seriously suggest you go and attend the mock test. Without attending the mock test, just by rigorous revision, many things will be lost. You will be evaporated, the things will be evaporated from your mind, and you will be saturated at the end. But if you try to attend at least a 1 or 2 mock tests every day, so that will give you a lot of confidence, and it will also build a capability that you can do well in your exams. That definitely helps. And also the one best thing about Agri Edit mock test is not only answering the mock test, you will be also getting the doubt clearance for whatever doubts you ask, for whatever questions that you attend. If you get any question, any doubt, you can directly post it in the community and get your doubts clarified. Okay, next one. So which of, again, seed testing part. See, till now, like if I have to recall, like we started to see the questions from floral biology, we started to see it from the questions from the seed physiology, seed certification, and seed testing. So these are the 4 categories from which the questions are frequently coming. So which of the following seed testing methods is used to assess the seed viability by artificially enhancing the age of seed? So now you will definitely ask me that, sir, you told that this is in the seed vigor, the test which have been mentioned here, which is accelerated aging test. Okay, this is the one which is artificially aged, but this was under actually seed vigor, but here they are asking about viability. So the students will definitely commit this mistake. As soon as they see the viability, they think that okay, for viability we have only one test, which is tetrazolim test, and they will mark the tetrazolim test. But please do not do this mistake. Even though in the books it is clearly mentioned that the accelerated aging test is for seed vigor, but in a closer sense you have to understand what is viability. See, whenever you do the seed vigor test, so what outcome you will have? So you will also get to know whether the seeds are alive or dead, and second thing, you will be also getting to know the potential of the seeds. Yes or no? Tell me. Like once you do the seed vigor testing, you will be knowing both whether the seeds are alive or not, and next thing you will be also getting to know the potential. But do the reverse. If you simply go for viability test, so will both the things will be addressed and answered? No, right? So that's why. So seed vigor is like the seed viability is also part of seed vigor testing, which we can also do it from the accelerated aging test. And whenever you see this artificially enhancing the aging, as you can correctly correlate here, this is the answer, accelerated aging test. Remember the term or the phrase that all vigorous seeds will be viable, but not all viable seeds will be vigorous. The viability will not exactly reflect the vigorness of the seed, But if the seed is vigorous, that means it is viable for sure. Remember this context. That will give you a good idea. Next, the following are the activities in the development and the release of the crop variety. Arrange the activities in a chronological order. So this I have talked in case of variety release notification chapter. I have discussed this in clearly. Let's see. Variety release and notification. Now, evaluation of the collected genotypes and selection of parental genotypes and collection of diverse genotypes for the target crop, pairwise hybridization, okay, testing, release and commercialization. So here you can definitely say the D will be last, and again in the options also D is last. So you have to make something out of it. So D is done, it will be last. Let us mention it as 4. The evaluation of collected samples of genotypes and selection of parental genotypes. So now first we have to go with this one, where we have to just collect the diverse genotypes for the target crop, and then we have to go for evaluation of the collected genotypes and selection of the parental genotypes. So out of collected genotypes, we will pick out some of the genotypes which are good for the parents, and then this is 1, this is 2, and then we will go for the pairwise crosses, that is hybridization. Third, and after doing it, we'll directly go for testing. If it suits good, we'll go for releasing and commercializing the variety. So the option is B, A, C, D. It is option B. So this is the answer. These things, like even if you study or if you read from the books or not, just by applying the common sense, you will be able to answer it. And in this video, I am not only discussing about the seed science. Whatever was not covered in the previous videos of genetics, previous videos of plant pathology and microbiology, and previous video of the plant breeding from the 2024 paper, all of them I have included here along with the seed science. Next thing, the one question again from the seed certification. Different classes of the seeds have various level of genetic purity standards. Arrange the following classes of the order ascending to the genetic purity. So as I told you, like I think I missed one thing while talking about the seed testing. I told you to remember working sample, I told you to remember the procedure, I asked you to remember the types, the reporting of results, but the standards I missed. So remember the standards for germination percentage for certification, genetic purity percentage for certification, and physical purity percentage for germination. These 3 are most important for your exam, that you have to make sure. And one more very important thing here is for germination percentage and physical purity percentage. So these things are same for foundation seed and certified seed. If I have to talk about germination percentage and the physical purity test percentage, then it is same for both foundation seed and certified seed. There is no difference. You need not to remember it differently, okay, separately. It is same germination percentage standard and the physical purity standard. It is same for both foundation seed and certified seed, but only for the genetic purity you have to remember separately for foundation seeds and certified seeds. So here they are asking the genetic purity standard for the different classes of seeds in their ascending order, and you know it already that it starts with the breeder seed first, and the foundation seeds, certified seeds, and the truthfully labeled seed. The breeder seed will be 100% pure, and what is the standard of foundation seed and what is the standard of certified seed, that is what you have to know according to the IMACS standard. What is IMACS? It is like Indian minimum seed certification standards, where the certification agency will follow the standards that is prescribed in this manual. And for the genetic purity, remember, for genetic purity there are different categories. For example, for the foundations we have 99% genetic purity is the standard, but again there is an exception which is for castor. Now can you tell me like why it is the exception? I want it in the comments, students. Whoever has listened to the class, I want it in the comments, like why the castor is a exception for it, where the genetic purity percentage is 95, for all other crops it is 99. Next, for certified, again for varieties, composite, synthetics and multi-lines it is 98%, and for hybrids normally it is 95%, but for certain hybrids of cotton, TPS, muskmelon, brinjal and tomato it is 90, and for castor it is 85. I also need the reason for this. I want you to comment on this video regarding the reasons why the genetic purity percentage for these hybrids is bit less than the standard one, which is 95. Okay, and also remember, I'll tell you one more thing, the hybrids which I mentioned, cotton, TPS, muskmelon, brinjal, tomato, castor, the 6 hybrids. So for these hybrids, generally the GOT is conducted in India for giving the seed certification, and we are not conducting GOT test for the varietal seed production. It is only conducted in the hybrid seed production, and also not for the hybrid seed production of all the crops. It is only for the 6 crops which are mentioned here down, for which the standards are slightly low, where we use the GOT for the genetic purity testing. Why? Everything I have already explained, so I want my students to explain this in the video on the comment. I would be definitely very happy to see the comments. Next one. So this is again the physical purity standards. If you are not aware about physical purity standards, and you might have read in the different books that different standards have been given, there is a lot of error in the numbers. So forget about every number, just try to remember this. So this is the accurate one. This is the exact standard that is mentioned in the IMSCS that you can remember. Okay. So this is for the physical purity. Next, similarly, like 2 things are mentioned. So what I try to compile here is standards which are field standards. Can you just recall the examples for the factors of the field standards? It is mainly the isolation distance that we know, and the specific factors can be like off types, inseparable other crop plants, or the plants affected by seed-borne diseases. These are the things for which we observe in the field. That's why it is called as field standards. So field standards, the standards are different for the foundation and the certified. So that is what I am trying to say. But when it comes to the seed standards, so they are same for all the factors. See, the pure seed same, inert matter same, the other crop seed same, the ODV, other distinguishable varieties, this is also same, wheat seed, germination, moisture content, vapor proof containers. For all these standards, the seed standards are same for both the foundation seeds and certified seed. The exception is what? Genetic purity testing. That's it. This is the one exception. Okay, this is the one example how we can remember it. For field standards, there is no difference in the foundation seed and the certified seed standard, but for the seed standards, for field standards there is difference between the foundation seed standard and the certified seed standard, but for the seed standards, there is no difference between the foundation seed and the certified seed. Hope it is clear. Okay, next, this is about the germination. Similarly, I talked about genetic purity, I talked about physical purity. Now it's about germination. So you might always get this question, sir, is it really necessary to remember all of this percentage? Will they really ask the example? Yes, they will ask the example, so you have to remember it. And it's very easy. See, you have to remember this, not too difficult. I won't say it is very easy, but it's not that difficult. If you revise it at least 4 to 5 times, you will be able to get this in the tip of your tongue. That's it. Very simple. Highest germination, remember, highest germination is in case of maize, lowest is 60 in case of capsicum and all gourds and carrot. And next you have 85, 80, 75, 70 and 65. Okay. Hope it is clear. So these things anyhow, once you get the PDFs, please make sure to write it somewhere and try to revise it regularly until you write the exams. That's important. Okay, let's go to the next question. Which of the following statement are incorrect about the enforced dormancy? Okay, it's again asking about the enforced dormancy, which we have looked at, which was explained in one of the previous systems, which was from the ARPR system. The enforced dormancy occurs due to the certain genetical and physiological factors. Okay, the enforced dormancy is truly not specific character of the seeds. Okay, and the high soil temperature and nitrogen content of the soil helps to break the enforced dormancy. Okay, the cultivation practices may also break the enforced dormancy, exposing the seeds to the better aeration. So what is the answer? First one, the enforced seed dormancy occurs due to the genetical and physiological factors. I told you it is like imposing the dormancy. Enforced, I told you this is because of the unfavorable conditions. So unfavorable condition itself means that it is because of the external condition. So it is not related to the genetic and physiological factors. And enforced dormancy is truly a specific character of seeds. Again, this is wrong. Since it is not genetical, it is not because of the inherent physiological activity. So this is not like heritable from generation to generation, and it cannot be like specific character of the seeds. So it is a general character. If you provide the unfavorable conditions, like all the varieties of the seeds will act in the same way. The answer is 3 and 4. That's it. Very simple. They are asking about, okay, incorrect. They are asking about incorrect. It is A and B only. So these two are wrong. So similarly, like what I did here, while reading the questions, make sure you read this part carefully. Anyhow, there was not a option of C and D. I immediately got remembered. But for you, while you write the exams, please make sure you remember and see this term incorrect. That's how you can answer it correctly. Otherwise, you will be missing it. Again, if you practice, you will be able to do it correctly. Which combinations of the following statements about the abscisic acids in the plants are correct? Okay. ABA is important for the plant development and desiccation tolerance. Okay. And ABA influences the expressions of various genes, including the gene for late embryogenesis abundant proteins, which are commonly called as LEA proteins. Okay. And ABA also regulates the closure of aquaporins. Fine. And like GAs, ABA also strongly promotes the flowering. Even if you do not have any idea about the ABA, do not have any idea about the seed physiological process, as soon as you see the last option, almost like everyone can answer this, say that this is wrong because it does not actually promote the flowering. That's for sure, right? So in this, see the options, all these are wrong and only first is right. For you, you have to answer like this only, because in the exam, do not think that even if you have 100% prepared for the exam, do not think that all the questions will be coming from the one which you have read. There will be questions where you have to just apply a common sense or a logic, you will get the answer. So this is one of that, but still I will be able to like break it. So ABA is important for the plant development and desiccation tolerance. If you remember the different phases of embryogenesis, it starts with the first stage, which is histodifferentiation, where the body plan is prepared, and mid stage, like early embryogenesis, first stage, mid embryogenesis, where the seed filling starts with the reserves, storage reserves, and in the late embryogenesis, if you remember, there is two different types of process which is occurring, which is called as desiccation tolerance and maturation drying, where there will be a drastic loss of water or the moisture content. But for this to take place, which is maturation drying, the drying to take place, the seeds should acquire an ability to withstand the drying, which is called as desiccation tolerance. So that is actually regulated by the hormone which is ABA. So this is correct. And next, the ABA influences the expression of the various genes. Yes, it is true, and it includes the LEA protein. So the hormonal regulation, because of the synthesis of ABA, it will actually activate the LEA protein, which acts as a molecular chaperones, which stabilizes the cell overall and try to help the cell to withstand the desiccation. So in a simple words, if you just go in depth, there are a lot of mechanisms that works here, but for you, remember these things, correlation between the LEA protein and the ABA. Because of the ABA, the LEA protein starts synthesizing, and this has a function as this functions as a molecular chaperones, which help in making the cell stable under the condition of drying. Okay. And third one, the ABA also regulates the closure of aquaporins. See, under stress conditions, you know this, right? You can relate with the stress and the ABA. Whenever there is stress to the plants or seeds, the ABA will be synthesized. So what it will do, as soon as the ABA is synthesized under stress, if there is a dot stress, then stomata should be closed or open. What do you think? Stomata should be closed or open. If stomata is open, there will be again evaporation, transpiration, again the water will be lost. So that should not occur specifically in case of dot condition. So in the dot condition, stomata should be closed, and this aquaporins, which are the water channels, so this will act on these channels, which will help in the closure, ultimately closure of stomata, which helps in maintaining the water level in the seeds or the plants, So that's how the ABA regulate this aquaporins. And the fourth option that I already discussed, it's not the one, wrong. So the upper three are correct. So for now, like the seed physiology part, like many of you might feel difficult because there is no standard book that from where you get all of this information. So what you can do is, when you read the seed physiology, please make sure to concentrate on three different things. So what are the three different things that you have to concentrate on? First one is seed development. So make sure to know everything about seed development. And second thing, concentrate on the germination, the process of germination plus the dormancy. And the third one, know about plant growth regulators, which are these hormones like ABA, auxin, cytokinins, ethylene and gibberellin. So all of this, just try to have a brief idea about its functions, its role, and how they are synthesized and how they are connected with the process of germination. Like for example, GA and AB are directly connected with the germination and dormancy. Just try to relate them and try to read them. So this is very much important, and mostly, like if you see, the questions will always come from this part in case of seed physiology. That's for sure. Okay. Next one. So again, the match the following type. So here we have completed the seed technology part, just left out with few general questions, general agriculture questions that we are going to deal. First one is, it's very simple to answer, like IRI, you know it, it's IRI New Delhi, like dairy, it's Colonel, and veterinary, it is Izzat Nagar, and the last one, the Fisheries Institute, it's in Mumbai. So it's like a boon, like you can easily answer it. Next one, National Insect Museum, where it is situated? This is situated in Bangalore. This is no trick for to remember. You have to like list all of the institutes of ICAR, all of the institutes which are related to agriculture, directly or indirectly, and try to revise it from time to time. Know its location, know its year, and for some of the important organizations like ICR Institute, like ICR and PPVFRI, also know the chairman, DG. Those things are important. Okay, number of national bureaus under ICR systems. So how many national bureaus, like how many deemed universities? So these things becomes important. Try to remember it. Here it is like 6. There are 6 national bureaus under the ICR systems at present. Okay. Next question. The questions we are doing right now, it's mostly related to the PGR. Okay. So the primary objective of the conservation of the plant genetic resources are to conserve the genetic diversity of the future generation. This is true. To promote the monoculture farming practices, wrong. To eradicate the invasive plant species, this is wrong. And to ensure the food security and the sustainable agriculture, okay, this is true. And to facilitate the research and the innovation in the plant breeding, this is also true. So according to this, the answer is A, D and E. Okay, this is the answer. Okay. Next one. So which of the following is not a serious limitation for the conserving of the plant genetic resources under in vitro repositories? Okay. So genetic, first one, the genetic stability may be compromised over the time due to the accumulation of the somaclonal variation. So this is true. See, when we are trying to produce or regenerate the plants from the tissue culture, if there is any variations, then that part which we are regenerating with the use of tissue culture, that will be of no use. So that's why this is a serious problem. And next, the high cost associated with the establishment and maintenance of in vitro. Okay, this is also true that the in vitro will definitely have the higher cost to bear, and limited capacity to conserve the large corrections compared to the field gene banks. Okay, and vulnerability to catastrophic events such as power outages and equipment. Since like we are doing it in artificial conditions, the power is again the most important one. But again, although all of these options seems that correct, but we have to choose one which is not serious limitation. It is a limitation but not serious. So if you just think about this, so that third one will be capacity. Although like capacity will be lower, agree, but there is not a serious limitation to use this in vitro technique. That's how we can conclude that third one will be the most suitable one, which is not a serious limitation for this in vitro method. Next one, the objectives of International Treaty of Plant Genetic Resources for the Food and Agriculture include. So it's again the objectives of this institute. So from this, what you can understand, please make sure to know the objectives of the international institutes and the different national institutes which are directly or indirectly related to the agriculture. And to promote the conservation and sustainable use of plant genetic resource, okay, good, true. To ensure the fair and equitable sharing of the benefits, okay, true. And to encourage the genetic modification of the crop plants and monoculture, no. and to facilitate the access of plant genetic resource for the food and agriculture, especially for the countries with the limited capacity. True. And to strengthen the capacity of the farmers, particularly in those developing countries, to conserve, manage and sustainably use the plant genetic resource. So apart from C, all of this is true. So the answer is A, B, D, E. Such questions, as I told you, like you have to apply the common sense. That's it, nothing more. So which of the following organization is primarily responsible for the collection, evaluation and conservation? So whatever we have discussed till now. First one, PPVFRA, like what is its main action plan? It is for the protection of plant varieties, right? So it mainly deals with the protection. And second, National Bureau of Plant Genetic Resources, where it is located, NBPGR. So it is located at New Delhi, just adjacent to the IRA campus. So this is the one which is true. Okay, fine. And this is Biodiversity International. It was earlier called as International Bureau of Plant Genetic Resource. So this was for the International Institute, but for here it is the National Bureau of Plant Genetic Resource, which is responsible for collection in India, for collection, evaluation and everything related to the plant genetic resource. This is the institute. So the second one is the correct option. Okay. So that's completes the 2024 PYQ analysis series from AgriAddict. So we started with genetics. We not only explained you or not only discussed about the options which is right, which you can also do it on your own. We also tried to add some value and try to discuss each one of the other options that have come in the exam, and try to explain it in a detailed way, so that if you watch these videos, definitely like most of the things will be very clear and you will be able to recall the things that you have already learnt. And also like if you have not learnt anything, you will be able to know like what is important and what is not. And I told you from seed tech, so what you have to focus on, like seed testing part, the different tests you have to focus on, seed physiology part that I just told you, seed certification part plus seed legislation. So these things are the most important one. Plus you can also get some questions from the seed processing regarding the equipments and its principle. That's it. That's it. Try to stick to these things first. If you have already covered these things, try to cover the other things. But if you have not yet covered these things, just stick to this. Just try to watch the 16 videos that I have uploaded in the YouTube. The links I will provide in the description box. So you can watch the 16 videos in a day for sure. You can do it in a day. That means like seed sense in a day. You can complete the seed sense in a day. So after watching the video, you will tell me definitely that I have covered most of the things and it is easy for you to read. And if you are already preparing for these exams, so the mock test will definitely help you. You can enroll our online mock test bundle which are currently going on. There are 50 plus mock tests which are available for the plant sciences. So links will be available in the description box. And as I told you, like along with the mock test, we also provide the doubt clearance that is much more useful at this last stage. And if you like our content and if you really want me to just start the marathon series to cover the separate subjects in a one go, then I need to encourage us in the comments, like, share and subscribe our YouTube channel. Thank you so much for watching this video and we'll meet in the next one.